In this screencast, I'm going to talk about heredity and genetics. And first we need to start with the father of genetics, which is this guy. You recognize him? Well, his name is Gregor Mendel, and that was a picture of him in 1870. He actually lived and worked about the same time as Charles Darwin. And as you can see by the cross on his chest, he was actually a monk, and he lived in a monastery in Austria. But he wasn't just a monk. He had a lot of education in math, and he understood a lot of agriculture. So he was really a scientist monk, if you can imagine that. And he worked with this plant right here, the pea, the flowering pea. And he looked at a lot of different traits in peas and did a lot of crosses. So for example, he took a purple flower and a white flower, and he knew how flowers, like humans, are sexual, and they have male and female organs. The males are called stamens, the females are called carpels, they have eggs in them. And he knew that peas tended to self-pollinate and produce offspring that looked just like themselves, kind of an asexual reproduction. But he said, what happens if I mix a purple male with a white female, or vice versa? So he actually started doing this and creating these hybrids. And whenever he did this, he found that he always got one of the traits. So a purple flower and a white flower produced all purple flowered plants. And he said, when I cross two of these traits, I'm always seeing one that is dominant. And he came up with this rule of dominance. Now we say it's a rule, not a law, because laws can't be broken and rules can. We'll talk about some exceptions to Mendel's rule. But for the most part, when he studied things, he saw that one trait was dominant over another. So here's the dominant purple flower and the white purple flower. And first off, when you look at it, it's purple and white. Well, we call that the phenotype. So the phen is what is seen. It's what it looks like. Purple versus white, or blue eyes versus brown eyes. Curly hair versus straight hair. That's the visible trait, okay? But what determines that trait is what's on the genes, what's in your DNA, and that's what we call the genotype. So the genotype is what's in your genes. That's the genetic makeup, okay? Now we're going to use symbols to represent the genotype. So let's look here for the purple. Since purple's dominant, we're going to say it's got the dominant allele. The allele is the variation. It could be purple, could be white. So since purple's dominant, we're going to always say big P for dominant, okay? But since it's dominant, it could be big P, or big P, little p. Because remember, we have homologous chromosomes, and we have these traits on those homologous chromosomes. If it shows the recessive, we're not going to say W for white, because we said the big P is dominant. So if it's recessive, it's just going to be little p. And because it doesn't show the dominant, it's got to have that little p on both of those homologous chromosomes. Okay? So Mendel, when he did this cross, so he crossed a purple and a white, and he got all purple, okay? Rule of dominance. But then he said, what happens if I cross two of these purple, right? Well, they should all be purple, right? Because purple's dominant. But that's not what he found. What he found was he got this ratio of about 3 purple to 1 white. So what's happening here? Well, that white isn't totally being dominated. It didn't go away. It's not like mixing paints, right? I mix purple and white. I can never get white paint again once I've mixed the two. But this is clearly coming back out. So he came up with his second rule. He said, if I cross two that are homozygous, homo means the same. So big P, and little p. I get one that I call heterozygous. Hetero means different. So I've got one big P and one little p. But the heterozygous, according to his rule of dominance, always shows the dominant trait. But if I cross two heterozygous, I get this ratio of about 3 to 1. 3 showing the dominant, 1 showing the recessive. His actual numbers were 705 and 224. He was pretty good at math, so we figured out that's pretty close to a 3 to 1 ratio. Okay. He did this with a lot of different traits. So purple and white, he always got purple showing the dominant over white, but when he crossed the heterozygous, he got that 3 to 1 ratio. He looked at where the flower goes. Does it go on the middle of the stem or the end of the stem? Axial versus terminal. Well, axial was dominant, but when he crossed the heterozygotes, he got, again, about a 3 to 1 ratio. Yellow versus green peas. Again, yellow is dominant, but the heterozygotes produced a 3 to 1 ratio. So for all these traits, he showed this exact same characteristic, that there was this rule of dominance, but there was also this idea of segregation, that the alleles segregated and passed on, and the recessives could still appear when we crossed heterozygous individuals. Okay? And again, the reason for that, going back to meiosis, is we know that meiosis segregates homologous chromosomes. So the alleles on the chromosomes could be for one trait or another. So we could have an allele for purple, or we could have an allele for white. Okay? If you have one of each, you're going to show the purple. But if you carry the white, you have a 50% chance of passing that white allele on to your sex cells. So this is why we see recessive phenotypes appearing in heterozygous individuals that show a dominant trait, because they'll still pass on that recessive allele during meiosis to 50% of their sex cells. Okay, so this gets to Mendel's second rule, which is the rule of segregation. So when I cross a dominant and a recessive, always show the dominant in what we call the F1, which is the first generation of offspring. P stands for parents' generation. Okay? But then when I cross those, my dominant and recessive alleles segregate and recombine in what's called a Punnett square. So if I have a big P, little p, I can see that about 1 out of 4 is going to get the little p. 2 out of 4 are going to get the big P, little p. They're going to be heterozygous and they're going to show the dominant. And then 1 out of 4 is going to be what we call homozygous dominant. It's going to show both of those alleles. So this is Mendel's rule of segregation. Even though a heterozygous individual shows the dominant trait, they'll still pass on that recessive allele 50% of the time. Okay, so that's Mendel's second rule. And again, sometimes we're looking at something that shows a dominant trait, and we want to know, is it homozygous or is it heterozygous? Well, the way I can figure that out is I can do a test cross. I can cross it with something that is homozygous recessive, right? If it's recessive, it has to be homozygous. It can't have the dominant allele. Okay? And the way I do that is I say, okay, if I cross it and it's homozygous, if it's pure dominant, all my offspring will show the dominant allele. Okay? But if it's heterozygous, if it's big P, little p, about half my offspring will show the recessive allele, because they will get that recessive allele. Again, the homozygous recessive will always pass on the recessive. So I can do this, it's called a test cross, to see if my individual showing the dominant phenotype is homozygous or heterozygous. Okay? So the last thing Mendel did is then he looked at different traits. So he said, okay, yellow is dominant to green, and round is dominant to wrinkled in P's. What happens if I cross a yellow round to a green wrinkled P? Okay, well, he got all yellow round, right? Because yellow is dominant and round is dominant. Okay. Then he said, what happens if I cross 2 of the F1 to produce the grandchildren, or the F2 generation? Okay, and so a lot of people would say, oh, well, you'll get 3 yellow round to 1 green wrinkled, right? You'll get this, but you can see the big X on there. That's not what you get, right? Because those 2 alleles segregate independently, and this is what Mendel called his third law of independent assortment, that all these different genes segregate and recombine independently. So this means when you're looking at a lot of different genes together, it gets really, really complicated, because there's a lot of different combinations you can get. So you can get the big Y and the big R. You can get the big Y and the little r. You can get the little y and the big R. You can get the little y and the little r. So just these 2 traits, if we do what's called a dihybrid cross, di meaning 2, hybrid meaning heterozygous for 2 crosses, we have a Punnett square that looks like this. And if we add them all up, we get this 9 out of 16 are yellow round, 3 out of 16 are green round, 3 out of 16 are yellow wrinkled, and 1 out of 16 is green wrinkled. So again, it gets really complicated, but this is because of Mendel's third law. It's called independent assortment. Because these genes are on different chromosomes, they don't just stay together like we see over here. They segregate and recombine independently. So when we're looking at a lot of different traits and the chances of them being passed on, it gets really, really complicated. And a lot of human traits are polygenic, which means many different genes govern them, and it makes it very difficult to predict exactly what the chances are that you'll see these in an offspring. So these are Mendel's big 3 ideas that he brought to genetics and heredity. And we're going to build on them, and we're going to look at some of the exceptions to Mendel's rules in the next couple of screencasts. But this is a good introduction to heredity and how Mendel helped sort of set the groundwork for our ideas on heredity and genetics.